Glycolysis. Glycolysis. Glycolysis is derived from the Greek words, glucose means sweet, lysis means dissolution. It is a universal pathway in the living cells. The complete pathway of glycolysis was elucidated in 1940. This pathway is often referred to as the Embden Meyerhof pathway, EM pathway, in honor of the two biochemists who made a major contribution to the knowledge of glycolysis. Glycolysis is defined as the sequence of reactions converting glucose, or glycogen, to pyruvate or lactate, with the production of ATP. Salient features of glycolysis. Number 1. Glycolysis takes place in all cells of the body. The enzymes of this pathway are in the cytosomal fraction of the cell. Number 2. Glycolysis occurs in aerobic and anaerobic conditions. Lactate is the end product under anaerobic condition. In the aerobic condition, pyruvate is formed, which is then oxidized to carbon dioxide and water. Number 3. Glycolysis is a major pathway for ATP synthesis in tissues lacking mitochondria, for example erythrocytes, cornea, lens, etc. Number 4. Glycolysis is very essential for the brain, which is dependent on glucose for energy. The glucose in brain has to undergo glycolysis before it is oxidized to carbon dioxide and water. Glycolysis in anaerobic condition may be summarized by the net reaction. Number 6. Glycolysis is a central metabolic pathway with many of its intermediates providing branch point to other pathways. Thus, the intermediates of glycolysis are useful for the synthesis of amino acids and fats. Number 7. Reversal of glycolysis along with the alternate arrangements at the irreversible steps, will result in the synthesis of glucose, that is gluconeogenesis. Reactions of glycolysis. The pathway can be divided into 3 distinct phases. A. Energy investment phase or priming stage. B. Splitting phase. C. Energy generation phase. Energy investment phase. Step number 1. Glucose is phosphorylated to glucose 6-phosphate by hexokinase or glucokinase, both are isoenzymes. This is an irreversible reaction, dependent on ATP and magnesium. Step number 2. Glucose 6-phosphate undergoes isomerization to give fructose 6-phosphate in the presence of the enzyme phosphohexose. Isomerase and magnesium. Step number 3. Fructose 6-phosphate is phosphorylated to fructose 1,6-bisphosphate by phosphofructokinase, PFK. This is an irreversible and a regulatory step in glycolysis. Splitting phase. Step number 4. The 6-carbon fructose 1,6-bisphosphate is split into 2 3-carbon compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase, fructose 1,6-bisphosphate aldolase. Step number 5. The enzyme phosphotrilyl isomerase catalyzes the reversible interconversion of glyceraldehyde 3-phosphate. Thus, 2 molecules of glyceraldehyde 3-phosphate are obtained from 1 molecule of glucose. Energy generation phase. Step number 6. Glyceraldehyde 3-phosphate dehydrogenase converts glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, with the production of NADH. Step number 7. The enzyme phosphoglycerate kinase acts on 1,3-bisphosphoglycerate, resulting in the synthesis of ATP and formation of 3-phosphoglycerate. Step number 8. 3-phosphoglycerate is converted to 2-phosphoglycerate by phosphoglycerate mutase. Step number 9. The high-energy compound phosphoenol pyruvate is generated from 2-phosphoglycerate by the enzyme enolase. Step number 10, the enzyme pyruvate kinase catalyzes the transfer of high-energy phosphate from phosphoenol pyruvate to ADP, leading to the formation of ATP and pyruvate. Anaerobic condition. In anaerobic condition, lack of oxygen, pyruvate is reduced by NADH to lactate in the presence of enzyme lactate dehydrogenase.